I'd like to welcome everybody to today's program that will provide an overview of the proposed exemptions for plant-incorporated protectants based on sexually compatible plants created through biotechnology. This webinar is produced by the U.S. Environmental Protection Agency's Office of Pesticide Programs. This is Mike Mendelsohn with the Biopesticides and Pollution Prevention Division, and I have the pleasure of being your moderator today. Before we begin, I'm pleased to welcome a distinguished group of presenters today, all of whom work at the EPA in the Emerging Technologies branch of the Biopesticides and Pollution Prevention Division. Dr. Eric Bohnenblust is a senior advisor who reviews pesticide registrations for biopesticides and efficacy data for public health pests. His past research focused on agroecosystem management using transgenic crops and sex pheromone mating disruption of codling moth and oriental fruit moth. Eric was the lead for development of these two proposed rules. Dr. Wiebke Striegel is a senior scientist whose education and research focused on plant molecular biology. Her training includes microbiology, genetics, and botany. And Dr. Striegel is one of the scientific co-leads for the development of these proposed rules. Dr. Amanda Pierce is a biologist who specializes in risk assessment and regulatory policy related to emerging technologies. Prior to her work at EPA, she earned her doctorate in ecology and evolution at Emory University, researching monarch butterfly population structure and migration. And Dr. Pierce is also a co-lead for the scientific portion of development of proposed rules. And I want to note that the comment period for these proposed rules runs from October 9th to December 8th. And the purpose of this webinar is for us to provide clarity and answer questions about the rule. But in order to make comments on these two proposed rules, please go to regulations.gov and do so that way. So as background, EPA regulates pesticides produced and used in plants. Plant-incorporated protectants are comprised of the pesticidal substance produced and used in the living plant and the genetic material necessary for its production. Also included in the regulatory definition are any inert ingredients present in the plant used to confirm the presence of the pesticidal substance, marker proteins, for instance, herbicide-resistant traits that are used to identify the presence in the transformed plant. Two overarching statutory authorities related to PIPs, or plant-incorporated protectants, are FIFRA, or the Federal Insecticide, Fungicide and Rodenticide Act. EPA evaluates pesticides, including PIPs, for their effects on the environment and human health and regulates their development, sale, distribution, and use. So FIFRA is the licensing statute for pesticides. Also, under the Federal Food, Drug, and Cosmetic Act, or FFDCA, EPA evaluates PIPs that are proposed for use in food or feed. In its assessment, the EPA considers all anticipated dietary exposures, residential and other outdoor uses. So FFDCA deals with residues in food and food safety. Historically, EPA has registered over 100 PIP products to date. The majority of these are Bacillus thuringiensis cryprotein-based traits for insect control. So going back to the definition of a PIP, in the BT example, the BT protein is the pesticidal substance, and the BT gene, often a cry or site gene, is the genetic material necessary for its production. These have mainly been in corn, cotton, and soy. We've also had some RNAi-based products. DV-SNF7 is an example in corn for corn rootworm control. It's the first RNAi approved for control of a macroorganism for the corn rootworm. Also, there are PIPs conferring disease resistance, viral coproteins, papaya and plum, squash, defensin proteins and for citrus screening, and also resistance genes like the R gene in the potato late blight product. The majority of registered PIPs to date were created using transgenesis of genes from organisms that are not sexually compatible with the recipient plant. Now, I'd like to talk about conventional breeding and PIPs that are already exempt from regulation. Since 2001, PIPs moved between sexually compatible plants through conventional breeding have been exempted from regulation under FIFRA and FFDCA. An exception to this is adverse effects reporting requirements are still required. So these are pesticide traits naturally found in plants that are bred into plants through conventional breeding. This exemption reflects the history of safe use of PIPs in conventional breeding to enhance pest resistance in new plant varieties. So these are, again, these pesticide traits in plants. The conventional breeding definition specifically excludes PIPs developed through biotechnology. That means that PIPs that are identical to those that could be moved through conventional breeding but are created through biotechnology currently require registration, even if they're indistinguishable. There's been an effort to update our regulations based on advances in biotechnology. Scientific advances now provide technologies to modify the genome such that plants modified using biotechnology can be indistinguishable from conventionally bred plants. In the Executive Order 13874 for modernizing the regulatory framework for ag biotech products, EPA was directed to use existing statutory authority as appropriate to exempt low-risk products of agricultural biotechnology from undue regulation. As part of the effort to implement this executive order, EPA has proposed these two rules. I also want to point out that the USDA recently revised its regulations at 7 CFR Part 340 through a rulemaking entitled Movement of Certain Genetically Engineered Organisms, better known as the SECURE rule. And this is also part of this overall effort. Now I'd like to turn the presentation over to Dr. Amanda Pierce, who will continue.
Thank you, Mike. So through this proposed rule, EPA has identified a new class of PIPs referred to as PIPs based on sexually compatible plants created through biotechnology. So through this proposed rule, EPA would exempt certain PIPs created through biotechnology under existing regulations, which are FIFRA and FFDCA, in the cases where those PIPs, 1, pose no greater risk than PIPs that EPA has already concluded meet safety requirements, and 2, could have otherwise been created through conventional breeding. So in other words, through this proposed rule, EPA aims to exempt certain PIPs created through biotechnology that are equivalent to those that are already exempt under the existing conventional breeding exemption. In addition to the exemption itself, the proposal also includes a process to determine eligibility for exemption. And there are two different routes here which are not mutually exclusive. The first one is a developer may submit a self-determination letter, and then either concurrently or subsequently, the developer may also request EPA confirmation that their PIP meets the criteria for exemption. Now, the general scientific rationale underlying this proposed rule relies on the framework that through our proposed criteria, EPA is limiting exempt pesticidal substances to only those that are found in plants that are sexually compatible with the recipient plant. Now, to achieve this, EPA has proposed some new definitions, including the definition for native gene and native allele. And these two definitions aid in limiting the pesticidal substances eligible for exemption to those found in plants that are sexually compatible with the recipient plant. Now, the use of the term native and native gene and native allele is intentional, as it is meant to specifically exclude the use of transgenes that could be moved between sexually compatible plants through conventional breeding. So here on the slide, we have an example to demonstrate what we mean by this. So here we have an example where you might have a bacterial endotoxin gene from BT that was engineered into a source plant. And although that source plant might be sexually compatible with the recipient plant, that BT gene would not meet the definition of a native gene because the BT organism is not sexually compatible with the recipient plant. So by limiting the pesticidal substance to only those that are found in plants sexually compatible with the recipient plant, EPA can rely on the history of safe use associated with conventional breeding to conclude negligible risk of novel exposures or hazards. So now I want to briefly describe some of the exemption categories under the proposed rule. The first of which would allow for modification of an existing gene to create a native allele or to insert a native gene. And this would allow for either modifications within the coding region, so long as those modifications ultimately create a native allele, and it would also allow for the insertion of a native gene, so long as that native gene is inserted into a non-genic region of the genome. The second category is differential expression of an existing gene. This category would allow for modifications in the expression level of an existing native gene, so long as it results in the production of the same substance. So in other words, one can change the expression either up or down of that gene, but the identity of the substance being produced must remain the same. And now this third category is loss of function of an existing gene. This one allows for reduction or elimination of a substance where that substance itself is not pesticidal, but the absence of that substance results in a pesticidal effect. So as you can see with these three categories, EPA's focus is on the substance, where that substance must either be identical to what was already present in the plant or what is identified in a sexually compatible relative. By focusing on the substance, that allows EPA to be flexible regarding the underlying nucleic acid sequence, as is demonstrated by inclusion of these differential expression categories. Now, the reason why EPA can allow that flexibility is because of this final bullet point, which has specific criteria limiting the expression profile of the pesticidal substance. Specifically, the pesticidal substance cannot exceed limits seen in sexually compatible plants. It also cannot differ from the expression pattern seen in sexually compatible plants. So in other words, it cannot be expressed in new tissues or in different developmental stages. And finally, of critical importance to the FFDCA exemption, the pesticidal substance cannot be present at levels that are injurious or deleterious to human health. I'd like to pass it off now to Wiebke Striegel, who will finish the rest of the presentation.
Yeah, thank you, Amanda. As Amanda already touched on, under the proposed structure, developers will have two options to obtain an exemption. They may either determine themselves that the PIP meets the criteria for exemption or request EPA review and confirmation. Importantly, these two paths are not mutually exclusive, and each path may provide certain advantages. For example, if submitted electronically, EPA response to a self-determination is immediate, and therefore the exemption goes into effect immediately. In this case, besides the general information of the submitter, we also require them to identify the specific exemption category that is applicable to the PIP. Now, on the other hand, for international trade considerations, for example, a developer may prefer EPA confirmation that their PIP has been reviewed by the agency and meets the criteria for exemption. What is applicable to the proposed exemption as a whole, and is the case for all PIPs, those exempt and requiring registration, any person who produces that PIP for sale and distribution is required to report any adverse incidences of harmful effects on humans and the environment to the agency. And therefore, under the proposed rule, EPA also withholds the right to revisit a specific eligibility determination at any time, and developers must maintain any records under the proposed record-keeping requirements. While EPA's confirmation process, by virtue of requiring review, would have a longer response time compared to the self-determination process, the request will be submitted under a specific Pesticide Registration Improvement Act category, or PRIA for short. That category is called M009, which limits the review timeframe by the agency to 4 months, and thereby increasing the predictability of the process for the developer. And again, the developer may choose to submit a self-determination and EPA determination request at the same time, which means that a PIP can already be exempt at the time a request for EPA confirmation is submitted. So what information is EPA requesting for an agency determination? There are 3 broad categories. Number 1 is the biology of the plant. For example, for a PIP that is created through a native gene insertion, this would include the identities of both the recipient and source plants, as well as information that the source and the recipient plant are sexually compatible. Second is information on the product, including what is the intended pesticidal function of the PIP and what pests are being targeted. Also of relevance is information to explain how the plant was modified. For example, if Cas9 was used, was it stably integrated into the genome of the plant? And if so, was it subsequently segregated out? This is important to understand whether there could be any PIP inert ingredients present in the final product, such as Cas9, which would not be covered under the proposed exemption and therefore may require their own tolerance exemption. In those instances in which the PIP is a known toxin that is commonly screened for in conventional breeding to ensure safe levels, one such example would be solanine in potatoes, we request confirmation that the developer similarly screens for the presence of the substance in their product. In doing so, this ensures that the pesticidal substance in the recipient food plant is at levels that are not injurious or deleterious to human health, as already mentioned by Amanda. The third broad category is the molecular characterization of the genetic change. Let's say an existing native gene is modified to produce the same pesticidal substance as a native allele. Then the nucleic acid and protein sequences will confirm that this is true. On the other hand, if the expression level has been changed, then the developer must show that the same expression patterns are maintained and that levels do not exceed those found in sexually compatible plants. This could be done either through a rationale, given the expected phenotype of the plant, or by providing a comparison of expression level data of the PIP in various tissues and developmental stages. An exemption is obtained for a specific PIP plant combination. That means that once an exemption has been obtained for a PIP through either of the means that I have just discussed, it is transferable to other plant varieties of the same plant species, as long as the submitter is doing one of the following. They either produce a substance in the other plant variety that is identical to the substance present in the exempt PIP, and no modification to the regulatory region of the modified gene is made, or they create the same phenotype in the other plant variety by targeting the same nucleic acid sequence in the regulatory region of the gene. In the proposed rules, EPA is also posing specific requests to the public that will inform the final PIPs rule. The 5 questions the agency poses are briefly summarized on the last slides of this presentation. First, EPA is requesting information as to which inert ingredients could be present in PIPs based on sexually compatible plants created through biotechnology. In the proposed rule, EPA discusses that the agency does not anticipate substances that would meet the definition of inert ingredient under 40 CFR 174.3 to be present in PIPs that would be eligible for the proposed exemption. Instead, EPA expects that potential inert ingredients be either segregated out during the breeding process or that they will only be transiently expressed as part of the initial PIP development process. Secondly, as mentioned earlier, in the event EPA learns of information that affects a previous determination that a PIP based on a sexually compatible plant created through biotechnology meets the criteria for exemption, EPA will consider the new information and provide a new determination in writing as to whether the PIP continues to meet the criteria for exemption. Now, the proposed rule does not specify the actions that would be taken by EPA in such an instance, and the agency would therefore request comment on whether additional details regarding this process would be beneficial to include in the final rule. Third, EPA is requesting comment on whether the agency should consider other approaches for determining the eligibility of a PIP for the proposed exemption and highlights 2 alternatives in the proposal. EPA requests comment on whether or how these alternative processes could be workably implemented and whether they are useful and justified. In one such approach, EPA could require the EPA confirmation process to be mandatory for all PIPs. This alternative process would follow the same submission procedures and information requirements that are already outlined for the current proposed EPA confirmation process in the proposal. Alternatively, the eligibility process could be made voluntary, which means that only those developers who actively seek EPA's confirmation would be required to submit to the agency information to establish that the PIP is eligible for exemption. EPA requests comment on whether the terms native and never derived are clear, or if alternative phrasing would be more appropriate. To illustrate how the terms are being used in the proposal, we have provided here parts of the proposed definition of a native gene. In that proposed definition, the phrase has never been derived from a source that is not sexually compatible with the source plant is meant to clarify that a PIP would not qualify for the proposed exemption if the gene originated from an organism that is not sexually compatible with a modified plant. In addition, EPA is requesting comment on whether the proposed definitions of native genes and native allele are necessary as a separate new definition, or if the criteria that define them should instead be incorporated into the exemption text. And lastly, EPA requests comment on whether the agency should issue a clarifying exemption for loss of function traits that result in pesticidal effects. Loss of function traits are those where the absence of a substance results in a pesticidal effect, for example, through a loss of a plant protein that would otherwise facilitate pathogenicity of a microbial pest. EPA considers these loss of function traits that confer a pesticidal effect to be a pesticide regulated under FIFRA, which in this case, the modified genetic material meeting the FIFRA 2U definition of a pesticide. Loss of function PIPs are currently included in the proposed exemption, but EPA requests comment as to whether a separate exemption would make the exemption status of these traits more clear, as unlike for the other PIPs encompassed in the proposed exemption, there is no pesticidal substance produced. To read the proposed rules for PIPs based on sexually compatible plants created through biotechnology, go to regulations.gov and search for docket EPA-HQ-OPP-2019-0508. And as Mike already mentioned at the beginning of the presentation, the public comment period is open until December 8, 2020.
All right, this is Mike Mendelsohn again. We're going to move into the questions. Before we do that, I want to mention a couple of things to clarify that this session is intended for the sole purpose of clarifying questions on the specifics of the proposed exemption. So EPA will not consider comments or suggestions provided during this session as public comments on the rule. Comments or suggestions on the proposed rule must be submitted to the docket during the public comment period for EPA to consider them in the final rule. And this presentation and any follow-up questions do not supersede the information that is published in the proposed rule. So I'll start off with a question here. Has EPA analyzed whether the scope of the proposed exemption is similar or different from the scope of other countries' regulatory frameworks, and what implications of any differences might have for international trade in food commodities? We did a lot of background work and tried to be as consistent as we could with USDA's secure rule. Again, we have different regulations that we have to implement. As was mentioned earlier, the Federal Food, Drug, and Cosmetic Act and the Federal Insecticide, Fungicide, and Rodenticide Act have certain requirements. And so that was a big part of shaping what we could do with the exemption to provide regulatory relief. But we did consider these things. All right, the next question is the self-determination letter made public? I'll turn that to Eric Bohnenblust.
At this time, EPA has not made a determination of whether there would be a website or anything like that, or whether we would specifically make it public.
The next question is, for indel type edits that result in a loss of function via the truncation of a coding sequence, does the exemption still apply since a non-functional truncated protein may be produced? Amanda, can you answer the question?
Yeah, so in this situation, if there's a substance that's being produced, that substance would have to be still identical to a substance that is in the plant already or in a sexually compatible relative. So in those situations, changes to the coding region would only be eligible for exemption if they still ultimately produce in a substance that's not novel to that plant or a sexually compatible relative.
Thanks, Amanda. The next question is, insertion of new genetic material using agrobacterium transformation can result in tDNA borders and cloning sequences being introduced that surround the gene. Question is, does the gene cassette from the donor plant have to be the same as introduced to the recipient plant? Or can the promoter terminator be from the donor plant but not associated with the gene of interest?
This is Wiebke. That's a good question. The way the proposal has defined it is that we are focusing on the gene itself. so the genetic material, including the regulatory and the coding region from the sexually compatible plant. So if the technique that is used can accomplish the insertion of that gene from the sexually compatible plant as the native gene, then that would be exempt under the proposal.
Thanks, Wiebke. Next question is on slide 10. It says injurious to health. Is there any analysis of whether the loss of function is injurious to animal health or harmful to non-targets?
So this is Amanda. So yes, the language injurious to health is specific to the FFDCA exemption, which is for tolerances and therefore is specifically looking at human health. But for the exemption under FIFRA, we did perform a risk analysis which looked at non-targets and determined that the PIPs that we're proposing for exemption would not pose any risk to non-target organisms relative to conventionally bred PIPs.
Thanks, Amanda. The next question, you presented that modifying an existing gene to create a native allele or insert a native gene allows for modifications within the coding region of an existing native gene in a plant to create a native allele. If a developer alters the coding region of a native gene to optimize expression, and that exact sequence is not found in a native allele, but the amino acid sequence of the protein has not changed, would this still qualify for an exemption?
This is Wiebke. Yeah, so long as the amino acid sequence of the protein is identical, one can modify the regulatory region to alter the genetic expression.
Thank you, Wiebke. The next question, what about polyploid crops? Are mutations in them covered by the sexually compatible exemption?
This is Wiebke again. Yeah, in this case, as long as the gene is found within the sexually compatible relatives that includes the source plant itself, then that would be covered.
Just to clarify, when you say gene, you mean the gene with the mutation?
Yeah, if we're talking about whatever is found in the plant, so yes.
Thanks, Wiebke. The next question on slide 12, number 2 mentions that screening of toxins and toxicants. Does that only apply to toxins that were the targeted trait, or is it applied more broadly with screening required for toxins not associated with the edited trait?
This is Wiebke. Since EPA's regulatory scope is on pesticidal substance and on pesticides, that would be applicable to the pesticidal substance produced through the gene-edited crop specifically.
Thanks, Wiebke. The next question, expression levels of proteins can vary drastically due to environmental factors. Has this been considered when determining the exemptions?
This is Amanda. Yes, that has been considered, which is why for the proposed exemption, one of the important pieces is always, in comparison to a sexually compatible relative, so that these PIPs created through biotech can be eligible for exemption when they're essentially equivalent to those that were already created through conventional breeding as well.
Thanks, Amanda. Next question. Will the presence of Agrobacterium border regions and other short cloning sequences disqualify the exemption of PIPs from sexually compatible plants?
We're not being specific in the proposal, and I would encourage, because that is an important question, I would encourage the submitter to submit this and underlying considerations to the docket so that we can give that further consideration and respond in writing, and so that everybody can see, also the people that are not currently on this webinar can see the response to that.
Thank you, Wiebke. Next question, would a modification resulting in a loss of function that includes modification of more than one allele be exempt under the new proposed rule?
If I understand the question correctly, for modifications of allele in potato, yes, so you can make several modifications in a particular plant.
Thanks, Wiebke. Next question, what guidance does EPA have on the types of information that would be considered adequate or inadequate to demonstrate that the donor plant and the recipient plant are sexually compatible?
This is Amanda. For this, I would encourage the person asking the question to also look under the definition of conventional breeding under 174.3 in EPA's existing regulations. If two plants are able to breed through any of these types of measures, then that would be an example of how plants are sexually compatible.
Thanks, Amanda. The next question is, slide 8 talks about comparing the exempt PIPs with already existing PIPs that pose no greater risk than PIPs that EPA has already concluded meet safety requirements. Is this referring to the PIPs on slide 5? How is the comparison made, and is it done on each PIP seeking an exemption?
This is Amanda. If I remember slide 5 correctly, I believe that was the one likely describing previous PIPs that have been registered by EPA, in which case those are not the PIPs that we're referring to. Specifically, the PIPs that we're referring to in our comparison are those that are exempt under the existing exemption for conventionally bred plants, so those that are sexually compatible with one another.
Thank you, Amanda. The next question, regarding the loss of function traits, because there is no pesticidal substance produced, and in some cases, to accomplish this, the entire genetic sequence, promoter, gene, terminator, might be deleted. What is the risk that EPA is trying to manage? And I'll turn that to Eric Bohnenblust.
In regard to the question, as far as what risks are EPA trying to manage, EPA assessed the risks associated with all of the PIPs that are proposed for exemption in the risk assessment part of the proposal. And our conclusion in the risk assessment piece is that these PIPs don't have any undue risk to humans or the environment under FIFRA, and as such qualify for exemption. In that sense, we believe they meet the statutory threshold for exemption, is what our proposal is showing. And so I think that kind of covers what we're looking at.
Thank you, Eric. Let's move on to the next question. There are tetraploid and diploid Gossypium species. Traditional breeding can cross a K diploid or bridging species to an A diploid, then to a tetraploid. Would the new rule exempt bypassing the bridging species to the tetraploid species?
What we're looking at is the sexually compatible framework here. So if it is possible through conventional breeding to create that bridging cross and introduce a specific gene into a recipient plant, then theoretically that would be covered under the proposal.
Thank you, Wiebke. Next question is, are alleles created through random mutagenesis considered native alleles? At full mutagenesis saturation, all possible genetic variations could be achieved, and therefore could not all possible genetic variations have the potential to be native alleles?
This is Wiebke. We would consider in the proposal alleles that are either created naturally through radiation or through means of conventional breeding. So in this case, as long as they're found in a sexually compatible plant, again, going back to this point, it would be considered part of the sexually compatible universe and therefore exempt.
Thanks, Wiebke. So they have to find it, or it has to be known to be present in the sexually compatible plant.
Yep, that's right. You would still have to have the comparator plant and the source plant available to support the rationale that this is within, the sexually compatible realm.
Thanks, Wiebke. All right, the next question is, the mutation in flavonoid synthesis will impact pigment production, the primary goal of selection. However, flavonoids are likely to be involved in defense pathways, not the goal of the selection. Will these be exempt? I'll turn this over to Eric Bohnenblust.
So I think what this gets at is the FIFRA statute and what is a pesticide versus not a pesticide under FIFRA. FIFRA is an intent-based statute, so anything that claims to have a pesticidal effect would be considered a pesticide. In the instance where you have a product and you don't have any intent to use it as a pesticide, and that isn't essentially the only use for it, that would not be considered a pesticide then. However, if you make claims that it would be a pesticide, then you would have to go through the exemption parameters to determine specifically if the change in question fits the exemption or not.
Thanks, Eric. All right, the next question. You mentioned the expression must not be higher than the native range. Could it be lower than the native range?
This is Amanda. Yes, the criteria is indeed that it cannot be higher, but then that means it can be lower and it can be all the way to 0, like as shown in the loss of function category.
Thanks, Amanda. All right, next question. Could you please explain the rationale behind the exemption of differential expression of a gene, but limiting it to expression profiles seen in sexually compatible plants with the same tissue in the same tissue or developmental stages? How could this be achieved in poorly described or studied genera?
The important distinction that we have here is that we, when crafting the proposal, are relying on a history of safe use of conventional breeding. And so the PIPs that are eligible for exemption must not result in novel hazards or novel exposures. And if the gene is modified so that expression is happening in a tissue that it didn't normally happen in, that could result in a novel exposure for non-targets, which is why we have to have these sort of criteria limiting that.
Thanks, Amanda. The next question is, for a polyploid crop, would deletion of the gene from all genomes count as a single edit or multiple edits? It seems that multiple edits do not have the potential to be exempted based on the proposed rule.
This is Wiebke. Yeah, you could, under the proposal, make the modifications in multiple genes in a polyploid species, and each one of them would still, given the other criteria for exemption are met, would still be individually exempted. So that is indeed possible.
Thank you, Wiebke. The next question is, are techniques such as embryo rescue and protoplast fusion considered as conventional breeding methods?
So since the proposal is exempting genes from plants that are in fact conventionally bred, and in some cases these techniques are being used in order to create the source plant, these genes would fall under the proposal of sexually compatible and could be submitted as something to support the rationale for sexual compatibility between the source and the recipient plant.
Thanks, Wiebke. So the next question is, will exempted PIPs be added to the FIFRA 25B list? Eric, if you could answer that.
I don't believe we've made a determination on how we're going to indicate what is exempt versus not exempt. So in that case, any comments on how we should handle that should be submitted to the docket.
Thanks, Eric. The next question is, has EPA reviewed any PIP products to date that would be eligible for the proposed exemption? Eric, if you could answer that.
We can't specifically provide that information, whether we have or have not at this time.
Thanks, Eric. The next question is for clarification. Are the exempted modifications for loss of function limited to those that are known?
This is Amanda. If your modification was going to be in the coding region and result in perhaps a partial protein, for example, that type of modification would need to be identified in a sexually compatible relative. But if your modification is going to be, say, in a regulatory region so that it's not changing the amino acid sequence of the gene, then that does not need to be identified in a sexually compatible relative. So for example, you could use like an SDN1 technique in the regulatory region.
Thanks, Amanda. A person wanted us to repeat the answer regarding public posting of the mandatory notification. Will EPA post any approved exemptions online? Eric, if you could answer that.
Yes, as I mentioned before, EPA has not made any determination on that. So any comments or thoughts that the public has on that should be submitted as comments to the docket for the rule, and they'll be considered in the final rule.
Thanks, Eric. The next question, PIP risk and toxicity on non-target organisms depends on concentration in tissues. What threshold does EPA use for exempt PIPs if the exempt PIP is re-engineered or reintroduced into a plant to produce at a much higher concentration than previously tested?
For this question, the important consideration is again our criteria limiting the expression profile of the exempt PIP. So by limiting the expression profile to only the same developmental stages and tissues, and so that it is not higher than what is found in a sexually compatible relative, we can still ensure that exempt biotech PIPs are equivalent to those PIPs that are already exempt under the conventional breeding exemption.
Thanks, Amanda. The next question is regarding the loss of function due to a gene deletion and no pesticidal substance being produced. What is EPA regulating in this scenario? And Eric, if you could answer that.
In that scenario, the claims made on the product determine the intent of the product. So if there's pesticidal claims made, then whatever the product is, the plant, the genetic material, as the case may be, or the deletion of the genetic material would be considered the pesticidal substance.
Thank you, Eric. Okay, the next question is, the proposal sets certain parameters with respect to expression: no new tissues, no new growth stage, and no higher level. Does the higher level limitation apply with respect to the growth stage and tissue? In other words, if the PIP substance in the donor plant is at 1x at growth stage 1 and 2x at growth stage 2, would a recipient plant that has a PIP substance at 2x at growth stage 1 be exempt?
That's a great question. So I would say that yes, it's intending to be relevant to the entire expression profile, but I would also encourage this questioner to submit their question to the docket so that we could also have it on record and have a formal answer as well.
Thank you, Amanda. The next question is, considering that expression level is not always correlated with protein concentration in plant tissue, and PIPs can translocate and accumulate in certain tissues, will this risk be considered in the exemption of a PIP?
This is Wiebke. Yeah, that's another great question, or two questions. In the proposal, we are making that point as well, that it is not always correlated, expression level with protein concentration, but generally that is the case in a general sense. Regarding the PIP concentration, or the protein, pesticide protein concentration, other tissues, if you consider the biology of the source plant, that would still translate from the source to the recipient plant. So you would still see that protein be translocated in the recipient and the source plant. So the expectation would be that the expression pattern would not be affected by the translocation of the protein.
Thanks, Wiebke. Okay, here's another question. It seems on slide 10 that an insertion of a native gene into a non-genic region would be exempted. Would such an insertion have to be done in a loss of function genetic background so that the expression level does not go outside the native range?
This is Wiebke again, another great question. So yeah, that is true. If an insertion of a native gene is made into a non-genic region, the criteria would be fulfilled for this proposed exemption. With regard to whether this needs to be done in a loss of function background, the main thing again is that the expression level should not exceed those found within sexually compatible plants. And as long as that criteria is fulfilled, the loss of function background is not necessarily required.
Thanks, Wiebke. Okay, the next question. If a loss of function gene is involved in a plant hormone pathway, is this gene-edited plant considered a PIP?
That's a great question. And that's one that I would encourage you to submit to the docket to get a written response to.
Okay, thanks. The next question is, if a person who is subject to the requirement to report adverse effects fails to do so, has that person committed a violation under FIFRA? And what, if anything, would such a violation mean for the status of the exemption? Again, Eric, if you could answer that.
If a adverse effect was not reported, that would be a violation under FIFRA. The specifics of what would happen would be determined by what the adverse effect was or what it would be determined to be. We can't speak specifically without a specific instance in that matter, but yes, that would be a violation of the adverse effects reporting requirement under FIFRA.
Thanks, Eric. The next question is, If an epigenetic change, for example, using methylation with no DNA changes, results in a pesticidal effect, would this be regulated or exempt as methylation occurs naturally in plants?
This is Wiebke. Yeah, that's a very good question. Again, we're not explicit in the proposal, as this really only deals with changes to the nucleic acid sequence. So that is one of those things that we would encourage to submit the question, and especially also the consideration underlying this question to the docket for the proposal for our consideration.
Thank you, Wiebke. Next question. Native expression levels are not static, but may change incrementally over time. How far outside the range can a level be in order to be exempted?
This is Amanda. One thing that would be maybe helpful in pointing out that Wiebke mentioned in her slides when she was describing the type of information for an EPA confirmation that would be required to demonstrate that the expression profile is the same. So if you didn't make any modifications in the regulatory region, then that alone would already provide rationale to support that there's no expectation that your expression profile would then be outside of what is seen in sexually compatible plants. So this type of information in terms of expression profile and which comparison to use is really relevant if you have intentionally modified the expression profile of your gene, in which case then yes, you would need to point to a comparator to illustrate that this is still within the bounds that's already found in sexually compatible plants.
Thanks, Amanda. Okay, the next question. Could you say more about the intended coordination with USDA's secure rule? Does EPA intend that plants exempt under this proposed rule should also meet the criteria for exemption under USDA's rule? I'll turn that over to Eric.
EPA and USDA did coordinate through interagency review prior to publishing the proposal. And as far as the scope of the two rules, there's as much similarity as there can be. Remember, we work under different statutes, so that dictates the scope of our different proposals. And so some things that are under EPA's rule would also meet the qualifications under USDA secure rule to be exempt by USDA as well. However, there may be things that EPA can exempt that would not be exempt under the secure rule and vice versa. In that context, the different statutes that we work under, as far as the Plant Pest Act and FIFRA and FFDCA, really drive what the different agencies can exempt. And that's why that even though there is as much similarity as we could get, there are some differences between the two rules.
Thanks, Eric. The next question. To be clear, do BT proteins identified in the first bullet on slide 5 fall under the proposed exemption?
Those would not fall under the proposed exemption, and that is because the BT proteins are derived from an organism that is not sexually compatible with the source plant, namely a Bacillus thuringiensis or any other organism for that matter. So although you could then cross subsequently a BT gene that is in a corn plant with another corn plant, that BT gene would not fall under the proposed exemption.
Thanks, Wiebke. Next question is, historically, how many applications for registration of PIPs has EPA seen that would be exempt under this proposal?
As far as the historical, what EPA has reviewed, we can't really discuss anything in-house that's not publicly known at this point. And so we can't disclose whether we have any applications in-house or not, or whether we have reviewed any other ones at this point.
Okay, thank you, Eric. Here's another question. Can exemption decisions change? For example, new native alleles will be discovered as more genome sequencing and more random mutagenesis populations are developed.
This seems to go to a question of whether once new alleles have been identified, if they would also be covered under the proposal. And yes, that would be the intent. This is not a snapshot that we're taking of the genetic diversity that is found within sexually compatible plants. So then if new alleles are found, then these would be considered as well.
Thanks a lot, Wiebke. Eric, are you ready to answer that earlier question?
Yeah, I can answer that now. As far as whether resistance management would be considered an adverse effect, in the risk assessment of the proposal, we don't anticipate any increased resistance risk posed by these PIPs based on sexually compatible plants created through biotechnology compared to those developed by conventional breeding. And we're not requiring specific resistance management plans from developers of these PIPs either. So from that, I would say that resistance would not be an adverse effect.
Okay, and I think for this question too, we just encourage the questioner to pose the question to the docket as well for a response and a response to comments. In the economic analysis, EPA estimates that only one new PIP containing product a year would qualify for the exemption criteria per year. If this number is 10 or 50-fold higher, would EPA be able to manage this workload? I will say that we have been in the process of increasing our capacity to handle increased workload. So we hope that we'll have enough to do the work, and if we get it even 50-fold higher, we'll do the best we can, but we have been increasing our capacity to be able to handle the workload. I think we're about ready to close. I just want to remind everybody that the session we just had with the Q&A was intended for the sole purpose of clarifying questions on the specifics of the proposed exemptions, and that EPA will not consider comments or suggestions provided during the session as public comments on the rule. Comments or suggestions on the proposed rule should be submitted to the docket during the comment period, which closes on December 8th, for EPA to consider them in the final rule, and we encourage you to do that. This presentation and any follow-up answers do not supersede the information that is published in the proposed rule. So with that, I'd like to thank everybody for attending our webinar today, and I hope everybody has a great day.